Welcome back to G Fun Facts Online. Today, we are not just talking about a scientific update, we are talking about a fundamental shift in how we interact with the building blocks of life. If you have been following biotechnology for the last 10 years, you know we have been living in the age of CRISPR. It has been called the magic scissors revolution. It earned Nobel Prizes, it launched billion-dollar companies, and it gave us the first real, tangible hope for curing genetic diseases. But today, I want to tell you why the age of CRISPR might be ending, or at least evolving into something unrecognizable. We are entering what scientists are calling the third age of genome engineering, and it is not driven by scissors, it is driven by a bridge, specifically a mechanism called Bridge Recombinase. To understand why this is such a massive deal, we have to look at the tool we have been using. CRISPR-Cas9 changed the world because it was programmable. That was the breakthrough. By changing a simple 20-letter sequence of RNA, scientists could tell the Cas9 protein to land anywhere in the genome. It was like a GPS for DNA, but here's the problem that nobody likes to talk about. Once CRISPR lands, its job is actually quite crude. It breaks things. It creates a double-strand break. It slices right through the DNA double helix. Now, the cell hates this. A severed DNA strand is a panic signal. So the cell rushes to repair the break. In doing so, it often makes mistakes, deleting a few letters or inserting random ones. This process, called non-homologous end-joining, is perfect if your goal is to break a gene, to knock it out. But what if you want to fix a gene? What if you want to insert a new sentence into the book of life? For that, CRISPR relies on a different repair pathway called homology-directed repair. But that pathway is inefficient. It works poorly in non-dividing cells like neurons, and it often fails, leading to a mix of edited and unedited cells. Furthermore, cutting DNA is dangerous. If you cut the genome in too many places, the chromosomes can shatter. It can trigger the p53 response, causing the cell to commit suicide. We have tried to fix this. We built base editors and prime editors, which are like chemical pens or word processors. They are brilliant, but they have a size limit. Prime editing chokes on large payloads. So the field was stuck. We could delete genes easily, and we could fix typos, but we lacked a reliable tool to insert massive sequences of genetic code into a specific location without risking genomic catastrophe. That is where the story of Bridge Recombinase begins. It starts not in a high-tech medical lab, but in the archives of microbial evolution. A team of researchers at the Arc Institute and the University of Tokyo, led by Dr. Patrick Shu and Dr. Hiroshi Nishimasu, were digging through the junk DNA of bacteria. They were looking at mobile genetic elements, often called jumping genes or transposons. These are selfish pieces of DNA that exist solely to copy themselves and move around. Most of them are messy. They jump randomly. But there was one family, known as IS-110, that was different. It was a minimalist. It seemed to cut itself out of the genome and paste itself into a new location with surgical precision, leaving no scars behind. The question was, how? How does it know where to go? In CRISPR, you have a guide RNA that holds the map, but IS-110 didn't seem to have one. Or so we thought. Using advanced bioinformatic mining and cryo-electron microscopy, the team found something that had been overlooked for decades. The IS-110 element encodes a non-coding RNA, but this is not a normal guide RNA. It is a structural marvel. It folds into two distinct loops. One loop binds to the target DNA in the genome. The other loop binds to the donor DNA, the cargo itself. It is a bridge. This RNA is the physical link between the donor and the target. It holds the cargo in one hand and the destination in the other, bringing them together for the enzyme to seal the deal. They called it the bridge RNA. This mechanism is a masterclass in molecular coordination. It is the first bispecific guide molecule found in nature. In the CRISPR system, the guide only knows the target. The donor DNA is just floating around, hoping to be used. In the bridge system, the bridge RNA physically tethers the donor to the target. It forces the interaction. The protein that drives this, the recombinase, is not a destroyer like Cas9. It is a constructor. It forms a complex of 4 units. 2 units grab the target, 2 units grab the donor, and the bridge RNA acts as the scaffold. Then comes the magic moment, the concerted strand exchange. The enzyme cuts all 4 strands of DNA simultaneously, but it doesn't let go. It holds onto the cut ends, swaps them, and re-glues them in a fraction of a second. There is never an exposed double-strand break. The genome is never left vulnerable. The DNA is simply rearranged. And the best part? It is programmable. The researchers found that the loops on the bridge RNA are modular. You can change the target loop to send the enzyme to any address in the genome. You can change the donor loop to pick up any piece of DNA you want to insert. It is a universal cut-and-paste adapter for the living world. Now, because this system relies on recombination, literally swapping the ends of DNA molecules, it offers a versatility that CRISPR struggles to match. It can perform 3 distinct topological operations. First, insertion. This is the holy grail. We often want to replace a broken gene with a healthy one. Bridge Recombinase can move megabase-scale sequences. We are talking about inserting entire gene clusters or synthetic pathways. Second, excision. Sometimes the problem is what is there, like toxic repetitions in Huntington's disease, or viral infections like HIV. The bridge system can loop out the bad section and seal the gap behind it. Third, inversion. Some genetic diseases are caused by a section of DNA that is flipped backward. The bridge system can grab that flipped section and flip it back. But of course, the big question was, does it work in humans? The initial discovery was in bacteria, but the graveyard of biotech is filled with tools that worked in bacteria and failed in human cells. The mammalian genome is complex. Patrick HSU's team didn't wait. They mined genomic databases for thousands of IS-110 relatives and found a variant called IS-622. In a stunning follow-up, they demonstrated that the IS-622 bridge recombinase is highly effective in human cells. They achieved insertion rates of over 20% for therapeutic payloads. In the world of knock-in gene editing, where 1 or 2% is often considered a success, 20% is revolutionary. They achieved over 98% on-target specificity, and they successfully manipulated nearly 1 million base pairs of DNA. This is chromosome engineering. One of the most compelling demonstrations involved Friedreich's ataxia, a neurodegenerative disease caused by a massive expansion of repeats in the FXN gene. These repeats act like a sticky trap, silencing the gene. Using bridge recombinase, the team successfully excised the entire repeat expansion block, restoring the gene's function. It was a clean surgery on the molecular level. To truly understand this, we have to look at the structure. Hiroshi Nishimasa's team used cryo-electron microscopy to freeze the bridge recombinase in action. The images revealed a dimer of dimers architecture. The proteins form a C-shaped clamp, and the bridge RNA threads through the center. This structure explains why the system is so specific. The enzyme is inactive until the bridge RNA binds both the target and the donor. It is a coincidence detector. It won't cut unless both conditions are met. This is a massive safety advantage over Cas9. The implications here extend far beyond correcting typos. This opens the door to synthetic genomics. Think about it. Current gene therapy is like trying to fix buggy software code by rewriting it line by line. Bridge recombinase allows us to upload a completely new, debugged version of the program. Instead of fixing a mutation, we can insert a pristine, full-length copy of the gene into a safe harbor location. In cancer immunotherapy, we can insert anti-cancer instructions into a precise location in T cells, ensuring they are potent and safe. For synthetic biologists, this allows for the rapid shuffling of gene orders to create more efficient biological assembly lines. Now, the bridge is still under construction. Delivery is a challenge. Getting megabase-sized DNA into a cell is physically difficult, and while specificity is high, it needs to be perfect for clinical use. But the trajectory is clear. We are moving from the age of the scissors to the age of the bridge. For billions of years, evolution was dictated by random mutations. CRISPR gave us the ability to intervene. But bridge recombinase suggests we are ready to move beyond intervention and into design. We are no longer just cutting the bonds of life. We are building bridges over the chasm of genetic disease toward a future where the genome is not a fixed destiny, but a canvas for human flourishing. Thanks for listening to G Fun Facts Online.